Hello, I'm Ryan Lim of Keystone Biology, and you're listening to Bio 700 Problem Solving course. This course consists of a total of 38 lectures, and in each lecture you'll find about 40 questions. So that gives you about 1,500 questions. That's tons of questions, right? But I didn't just put in all the random questions. Those questions were carefully selected and revised from the real estate questions that I've compiled over the past few years and from really important books. So needless to say, after perfect completion of this course, your score will boost up a lot, right? However, there's one condition. You need to solve the questions at least 3 times, all right? And the best and yet easiest way to do this is by making your own notepad with the questions you got wrong or didn't know. So I'm going to give you a little tip on how to make this. First, you just print out the whole lecture, maybe 4 slides per page, and then solve the whole lectures twice or 3 times. And make sure you do not write answer on it, but instead just to write the explanations on it so that you can know why you got this question wrong and what was the point that you didn't understand, right? And then either cut and paste or copy down the questions on your notebook and solve the notepad multiple times. That's what you need to do. So as you're drawing near to the test day, you'll get more and more confident because the only thing you need to hold on to is these 2 little notebooks. First one for your main lecture, the review notepad, and the second one for this problem-solving course, right? In my past experience, over the few years, I've never seen a single student who've got over 700 without the 2 notes. So make sure you make it, okay? Because this course, biology, never betray the time and effort you put in, all right? All you need to do, trust me, and follow my lead. All right, if you're ready, let's get started. Okay, lecture 1. It's on biochemistry, and it's section 1. We have more sections on biochemistry. Okay, let's get to number 1. What happens to an enzyme during a biochemical reaction? It's about enzyme, right? On exam, I guarantee you're going to see at least 1 or 2 questions on enzyme. And what's the most important thing about enzyme? This is organic catalyst, right? Organic catalyst. And catalyst, by definition, is something that doesn't change, does participate in a reaction, but themselves, they never change, right? Okay, A, it becomes part of the product. Nope. It implies the change of the catalyst, right? B, it is unchanged. Yes. Right here, we have our answer. Okay, 2, this graph shows the energy profile of certain reaction. Which graph shows the reaction when enzymes are added. And this graph, I'm pretty sure you've seen this like millions of times, right? Here you have free energy on the y-axis. Here on the x-axis you have a reaction progress or time here, right? So this is the energy level of the reactant, and this is the energy level of the product, right? But either equilibrium or the energy level of the both reactant and the product can never change. It can be never changed by the presence of the enzymes. So these 2 levels should be kept exactly the same, but it's just this much, this energy field that should be changed. What is this? This is activation energy, right? The amount of the energy you need to overcome, to complete, or push off this reaction from reactant to the product part, right? So if the activation energy is lowered, you're going to have a graph like this, right, with much lower hill. So That's what we're going to find from the options. All right, here, no, the energy of the product level changed, reactant. And this one, too high, never going to happen. Yes, D. Here we have our answer. Okay, 3, a pure sample of some sort of macromolecule is obtained after complete hydrolyzation of the molecules. Nucleotide uracil is detected. So obviously, what we are having for the macromolecule is a nucleic acid. So we have 2 options. It could have been DNA, could have been RNA, but since we found uracil, it can never be DNA. So we know it is RNA. All right. A, it is a nucleic acid. This is true. B, it is formed using a dehydration condensation reaction of individual nucleotides. Correct. Either the protein, all the macromolecules are made by dehydration synthesis or dehydration condensation. C, it contains a sugar phosphate backbone. Of course, both the DNA and RNA has this. D, it generally exists as a double helix. Nope. This is a configuration that can be only found in what? DNA. So D is wrong statement. Okay, for a solution with a pH of 5 is how many times more acidic than the solution of pH 5, pH 7? Okay, pH is 5. That means minus log, put on the concentration of the hydrogen is 5. So you have a 10 hidden here. So that means this is the concentration of the hydrogen. And if the pH is 7, this is the concentration of hydrogen. So if you compare these 2 numbers, you know you got to have 100 here. So D. Okay, 5. Which of the following is a polymer? 1. Hemoglobin. What is it? This is a protein that can be found within red blood cell or erythrocytes, right? So protein is obviously polymers of what? Amino acid. So 1 is right. And cellulose, we know this is a polysaccharide, right? So 2 also. But steroids, what is it? Steroid is just one of a kind of lipids, right? For the lipids, you can have a fat, you can have a phospholipid, you can have a steroids. And steroid is just one of the kind of the lipids that contains 4 fused rings like this, right? 1 ring, 2 ring fused, and another ring fused together, and the last 4th ring like this. But they usually exist in monomer, just 4 rings. That's it. They don't make polymer. All right? So because of that, steroid is canceled out. What you got is 1 and 2. So that's why the answer is C. Okay, question 6 to 7. I know this is phospholipid because you have 2 tails, 2 fatty acids, which is made of the hydrocarbon. So this is basically hydrophobic region. And what you have here is, along this one, glyceride. Okay, glyceride, and this part on the top of the glyceride, phosphate group, or just simply Pi. Right? So let's see what questions are asking. 6, which of the following describes the molecule above? It is phospholipid, right? Okay, A, it is made using a glycosidic bond. Nope. Glycosidic bond is something, chemical bond used to make polymers of the polysaccharides. So nope, it is not a polysaccharide. B, it provides a template for protein synthesis. Nope. It's talking about mRNA. It is a nucleic acid. C, it is a type of steroid. Nope. Type of lipids. D, it is an amphipathic molecule. Yes. What does amphipathic means? Amphi means like bi on the both sides, right? So